You all must be wondering that why the term DNA fingerprint came into existence, because it has been adopted from the real fingerprint, which was used for hundreds of years to have the uniqueness of every individual. In fact, if you see the image, that you would really wonder that how it has been invented that every individual has a very unique DNA fingerprint. In fact, in the year 1858, Sir William James Herschel was a chief magistrate of Hooghly district in Jangipur. He was the one used this on native contracts because of the language which they cannot understand, and he was in a fear that whether they may take away money or property. So in order to have some records, he has used that. But what is even more interesting is that he didn't leave this, or his colleagues did not leave that, and they have observed that for several years. And in fact, after 160 years, so they have published this paper in Nature. You can imagine that a non-scientist publishing his observation in a topmost journal like Nature. Okay, so then it gives the authenticity of the technique. But when Professor Alex Jeffrey, when he was working on his basic research, and suddenly he found that when he hybridized the human DNA with a class of repeats from beta-globin gene, and he suddenly found that every individual has a very unique DNA fingerprint pattern or DNA pattern. So later on, he named as a DNA fingerprinting. So this is how every individual's uniqueness has been observed. So subsequently, he has used this for several cases. The same year, he published this detail in 4 different papers in the journal Nature. So while this was going on, we must be happy that so India became the 3rd country to develop its own probe. After Alex Jeffrey published this paper, and Dr. Lalji Singh was working on his basic research in the area of sex determination in snakes. In snake, there are 3 different kinds of snakes: primitive snake, intermediate, and highly advanced. In case of human, we have 46 chromosomes, and 44 chromosomes are common between male and female, and only pair of chromosome is the sex chromosome, that's X and Y. In case of female, there will be 2X, and in male, 1X and 1Y chromosome. But in case of snakes, it's called as WZ chromosomes. Then the primitive snake, in human, you can see that X is a long chromosome and Y is very short. But in case of snake, the primitive snake will not see any difference between this Z and W chromosome. But in the intermediate snake, also you will not see any size difference. But there's accumulation of this GATA repeat, what you see in the red color, GATA as a tandemly repeated. But in case of the highly evolved snake, you can see the difference between W and Z chromosome. So this was his basic research. Once the data was published by Alex Jeffrey, then he thought, let us use this and see whether this probe also generate the individual specific DNA fingerprint. And interestingly, he also found that not only in human and many other species, that is giving the DNA fingerprinting pattern, mostly they are individual specific. And this is one of the historical historical slide where you can see that the very first case where he himself went and gave the evidence in the court of law. There's a case from Kerala, Tellicherry, where he's a contractor, and he convinced her that he will marry her, and she became pregnant and the child was born. Then he refused to marry her, saying that this child is not belongs to him. So that's where she approached CCMB, and she read about DNA fingerprint technology, and the sample was analyzed, and the data, the report was given, and Dr. Singh himself went to the court in Telacheri and gave the evidence. So historically, so this is the very first case where DNA fingerprinting evidence was produced in the court of law. And in fact, the center which I am heading, Center for DNA Fingerprinting and Diagnostics, have emerged from there, and he visualized, and Dr. Bhargava was the then director of CCMB, all thought that there could be a separate institute for tackling this issue, providing DNA fingerprinting and also diagnostics of various human diseases. That's how the Center for DNA Fingerprinting and Diagnostics have started, and we are at the 25th years. We are celebrating the Silver Jubilee. For the non-subject participant, just wanted to take you through the basics. I mentioned that in human, there are 46 chromosomes. In the nucleus, there's a 46 chromosome. In somatic cell, that means all the body cells. Of course, the germ cell, there'll be half of it, 23 chromosomes. So this is called as nuclear genome. In addition to that, so this is very important. In the coming slides, I'll tell you. So there is a organelle called mitochondria. It's generally known as a powerhouse of the cell, because that's where the ATP is produced. In addition to that, it has a large number of applications, the function. In a given cell, there'll be more mitochondria, and in a given mitochondria, there'll be large number of mitochondrial DNA. So in a given cell, you will find 2 copies of the nuclear genome.
Apologies, Dr. Thangaraj, you can continue.
Sorry, you got muted. Can you unmute yourself?
Sorry. Let me go to the previous slide. Yeah. In a given cell, as I was mentioning, 2 copies of nuclear genome, whereas in the mitochondria, so there will be in the mitochondria, there will be large number of mitochondrial DNA, hundreds of mitochondrial DNA. It has its own application, which I'll tell you later. But it's also important to understand how the genome is inherited. For example, somatic cell, I said there are 46 chromosomes. This is the female karyotype, where there are 2 X chromosomes in addition to 22 pairs of autosome. This is the male karyotype. In addition to 22 pairs of autosome, there's 1 X chromosome and 1 Y chromosome. The Y chromosome is the one which consists of gene called SRY, which is essential for determining the male sex of the individual. Okay, these autosomes are common. There's no difference between male and female. But when the meiosis, when the cell is dividing in the germ cell, then there will be only half of that chromosome goes into the germ cell. So where you have 23 pairs, and here in the germ cell, if you see in the egg, only 23. Okay, 46 again this 23. So that means there's a reduction of the chromosome. Same way in the male, in the sperm, you'll have only 23 chromosomes. But the division of this, the segregation of the chromosomes are not well defined. It's a random. So that's why every individual, for example, this chromosome from here, you see that there could be chromosome 1. This from this pair, only 1 of this goes there. From here, this chromosome goes there. But in the next cell division, instead of this may go with the combination of this. So that's the reason every individual, even the family, all the six, there are even 5 or 6 or 8 son or daughter, every individual is different, except for the monozygotic twin, because they will have exactly the same genome. Okay, so the important point here is that the egg will carry only half of the genome and the sperm will carry only half of the genome. At the time of fertilization, become 46 chromosomes. That means 23 chromosomes goes from the father, 23 from the mother, or 50% of the genome from father, 50% from the mother. Whereas look at that, the mitochondria, which I was mentioning, the egg will have the mitochondria, whereas in the sperm, the mitochondria will be the mid piece of the sperm. At the time of fertilization, it remain outside. Even if it goes, then it will get degraded. Therefore, we all get the mitochondria, mitochondrial DNA only from the mother. So it's maternally inherited. So that's a good point to remember. If you talk about some of the fact about the DNA, it's very stable. If you eat it, it will become a single strand, because DNA is double strand. And if you allow it to cool down, become double strand again. And it can be isolated from any biological material, blood, any part of the tissue, saliva, sperm, or air root, bone, any biological material. So saliva, urine, air, teeth, bone, any kind of tissue. Okay. And so at every exchange, so if you talk about the forensics, when somebody touch or drink coffee or smoke, so you'll find the epithelial cell in wherever they touch or wherever they tea, coffee they drink on the rim of the glass or teacup. Okay, so that gives valuable sample to analyze and get the DNA profile. And it doesn't change with the age. If you take DNA of a just born baby and generate profile, or you wait till 50 years or 60 years, again generate the profile, you will not see any difference. There will be some difference except in case of, for example, cancer. So the cancer tissue might undergo a lot of rearrangement. Only there you might see some variation. Otherwise, generally you don't see much of variations. And the pattern of an individual from every tissue, if you take blood, if you take tooth, if you tooth pulp, in fact, or if you take air root or bone, if you generate the profile, all of them should be identical. And most importantly, it shows variation between individual, because we have huge amount of variation. If you take single nucleotide polymorphism, you will find, although we are all having the same genome and we are up to 99.6% similarity, but only 0.4% difference between individual. But that 0.4% actually gives 4 million differences between individuals. So, so that is very, very important when you do DNA-based analysis. Shows a lot more variation. So if you look at the variations, I'll not go into detail about the chromosomal level, but if you look at the DNA level variations, you'll find there could be a tandem deletion or duplication. Okay, as you see here. So there's a duplication of BC, right? So BC and become a BC. So there could be a deletion, there could be an inversion from one direction to other direction. That happens in megabase level. But if you come down to kilobase level, you'll have ALU element insertion. There's a mini satellites, line one, and there are many repetitive sequences. But if you come down at small stretch up repeat, like short tandem repeats or STRs, what you see here, CT80, that repeat for many times. In one chromosome may have some 10 repeat, other chromosome may have 15 repeat or 20 repeat. But if you come down to single nucleotide level, there could be, as I mentioned, about 4 million difference. As you see here, there's a the sequence is common between these two sequences, but only difference is here at the single nucleotide level. Here you see there's a C, and whereas in the other sequence there's a T. So that makes the difference, right? And when the DNA fingerprinting was developed, it was based on the southern hybridization method or RFLP based method. That's the restriction fragment length polymorphism. And if you take two individuals' DNA, and if you use restriction enzyme, as all of you are aware that each restriction enzyme will have a specific motive. For example, if you use EcoR1 as an enzyme, it recognizes wherever there's a GAATTC. So it cuts in that position. Okay. So if you take these as an example, two different fragments, there is one EcoR1 site, this is another EcoR1 site, this is a third EcoR1 site. Okay. Here, there's one EcoR1 site. Here, there is a single nucleotide polymorphism variant. Instead of there is a, there is a G here. Okay, A to G variation. So, so in this case, when you use EcoR1, it will cut the DNA at this position, and this position. So you will get 1, 2, 3, and 4 fragments. Whereas here it will not cut because there's a change in the nucleotide. So EcoR1 will not recognize that as a motive, so that it will not cut here. So instead of these two fragment, the individual who has this kind of variation, they'll get only one band. So that's how the variations are generated. So this is a process. Once you have a DNA isolated, and that will be treated with the restriction enzymes, therefore the DNA will become fragmented. Then that would be used to separate as per the size of the DNA fragment using gel electrophoresis. Then that will be transferred onto nylon membrane because the gel is not stable. So while transferring, you make the DNA into single strand because DNA is double strand, treat with sodium hydroxide. Then the nylon membrane will have the DNA, which is in a single strand. Then you take a DNA probe. In this case, it's easy to remember, GATA repeat. Use this as a probe, but label that with radioactive material. Therefore, wherever it finds its complementary sequence, then it will anneal and it stick to that, become a double strand there. But it, at the end of it, it has the radioactive material. Therefore, when you expose that onto X-ray film, it emits the radiation. Therefore, you will see after developing the film, you will see that band. That means these are the place where the probe which we use, actually DNA, when you run, it will initially show as a smear. There will not be any band. So once you hybridize with the probe, which is labeled with radioactive material, it hybridizes, and after a few days, once you develop it and it gives a band, that means this is the place where the probe which you used have hybridized. Okay, then you can see that each individual shows very unique banding pattern. That means, so each individual has different, depending upon the restriction enzyme used. For example, EcoR1, if you take, that means each individual have different position. Somebody might have the restriction site, somebody may not have the restriction site. So that's the reason it is giving banding pattern like this. So now we come to the STRs, because these are the technique which was developed earlier, but is not being used because it's very, very time consuming and it takes several days, at least about 1 week to 10 days to get it. But now it become very simple PCR based method. So where you have the repetitive sequence, for example, here, there are 3 different kind of repeats. It's a CT repeat, dinucleotide. This is a CAG repeat, trinucleotide. And this is a tetranucleotide repeat, CT8. There is no different. There's no interfering sequence here. They're all tandemly repeated. But on both the sides, flanking to this sequence, you have very unique sequence. So one can design primer on the both the end of the repeat, then use that as a primer to amplify. For example, somebody has 15 repeat here, other person has 20 repeat. So there will be a small band and other one will have a bigger band depending upon the size of the repeats, which I'll tell you in coming slide. Here again, it's very important. for understanding how the DNA fingerprinting can be interpreted. For example, I was mentioning earlier that we have inherited 50% of the genome from father, 50% from the mother. Okay, so this is what is shown here. Let's look at here. Okay, this there are 3 seeds. This is the pedigree, father, mother, and their 3 children, 1 son and 2 daughters. Okay, let's take here, and he inherited. You consider this as one of the autosome, 50% from the father, and the remaining 50% from the mother. And in this case, she inherited 50% from the father and 50% mother. And this is, you assume that this is a mitochondrial DNA. There's no paternal contribution. So all the mitochondria, all the 3 have inherited mitochondrial DNA only from the mother. And of course, about Y chromosome, I didn't mention earlier, although I mentioned in the beginning, but the Y chromosome is present only in the male because that is essential for making individual as a male. So that you will find only in the male, and you can see all the males will have the same chromosomes inherited as such, because it doesn't have homology anywhere except in the pseudo-autosomal region. Therefore, it goes generation after generation without much change. Okay, so this itself will give you some clue. And on the right side, if you look at the DNA profile, the actual DNA profile, now you can imagine 50%. Okay, so that means if you take one particular locus, one region you are amplifying, and trying to see whether it's matching, one band is matching with the father, one band from the other, because this is 2 copies, right? Let us look at this particular locus, the one blue one. Okay, this is mother's profile, the first lane. This is sons, and this is this is sons, and this is fathers. Okay, if you take this locus, the son has 2 alleles. Okay, in a locus, 2 alleles. So of that, 1 allele has come from the mother. Okay, consider this. Okay, this has come from father. Let's say this has come from the mother. Okay, so it's matching with the mother. Other one, it has come from the father. Okay, so this green one. Okay, same way, if you look at here, one band is matching with the mother, other one is matching with the father. Okay, same way, all of these. If you look at every single locus, then you can actually precisely tell whether he is the father of the child or she is the mother of the child. Okay, so that precise one can able to say, because this is nothing but the variation, nothing but the repeat number present in that particular locus. Okay, so the longer one means this has a more number of repeat, shorter one has a very small number of repeat. Sometimes you may see only one band. Okay, that's a homozygous. That means both father and mother have the same allele and contribute to the same allele. Okay, so this is the basis for anybody. If you have doubt, we can discuss towards end of my talk. Right? On the other side, you can again take this particular locus. Okay, same locus, but different individual. Now you see, again, this is a mother, father, and the child. Now there are 2 alleles. One allele is matching with the mother. This allele, if he is a father, he should have either matched with this or matching with that. Same way here, one allele is matching with the mother, and this allele, if he is the father, he should have been either here or here. Okay, there are many which wherever there's a arrow mark. So you can precisely say 99, statistically 99.99% precisely you can say whether he is a father of the child or she is a mother of child, or this child belongs to this family or not. So there are many ways of interpreting. So this is the very straightforward STR profile which can establish the identity of the individual or paternity or maternity of the individual. So what is the advantage of doing this? I already mentioned that it requires very small quantity of DNA. Even very highly degraded DNA also can be used because you are amplifying very, very small region. And you can get the result in 24 hours, multiplexing, as you see in several bands, and one can do automation. And this is what being used universally all over the world. This is a standard STR kit is used for forensic application. So now let's look at very briefly about the characteristic feature of the Y chromosome. So when it is used, when the father is not available, one can use any of his paternally related individuals. So almost all the individuals in the family, they are paternally related, will have the same profile. And tracing the paternal lineage used, you want to identify the male DNA, you can use, and there is a mixture of male and female DNA, you wanted to precisely identify the male DNA, you can use, and the paternity without mother sample. Because of lack of time, I'll very briefly I'll not go into detail about that. And this is again the profile generated using autosomal marker, where you see that father and the son having the same STR profile. That means there's no difference in the Y chromosome. There's one Y chromosome, it went as such from father to son, then it go to grandson. And sometimes you have the mixed sample, can able to identify how many individuals actually involved in the crime. So this is the autosome. It's very difficult because a lot of alleles are mixed here, but this can tell you that how many individuals. So this is one individual, this is another individual, the third individual. So one can able to make use of this for precisely identifying how many individuals and who are the individuals involved in the crime. And this, I must say that how historically that Jefferson was the third president of US has been identified as a real father of one of his slave son. So if you look at this slide, that you'll be able to understand how old the case is. And he was he lived between 1743 to 1826. He was the third US president. He had a slave called Sally Hamming, and one of her son, Easton. He resembled phenotypically with the Jefferson. But what happened that there was a lot of review about that, and about a decade ago, or maybe little more than a decade ago, there are three teams. One from Oxford, Chris Taylor Smith, from Leicester, Mark Javelin, he was a colleague of Alec Jeffrey, and Peter Denif from Leiden. So they joined together and used several Y chromosome markers and try to understand whether Jefferson is the real father of Easton. So they used different kind of marker. This is a biallelic marker like SNPs, A or G or C or T, something like that. These are microsatellite markers or STR markers, which I was describing. And these are mini satellite markers. There was no immediate lineage of Jefferson because he had one son and he died in the car accident. So they used his paternal uncle's lineage. This is President Jefferson's father's brother, and each number represents one generation. So that means they are trying to identify whether Jefferson is the real father of Easton after 6-7 generations. You can see the power of the Y chromosome marker. So then this is the Easton's again lineage, and these two are another suspects. You can compare that with the color code which they have published this paper. You can see that except there's one mutation, otherwise this profile is exactly the same as this lineage. These are all different, and they concluded that yes, Sally Hamming's son Easton, the Jefferson was actually father of Easton. You can imagine after several generations. So of course, we use this technology for tracing the human origin and migration. So we also identify the Andaman tribes have migrated from Africa about 65,000 years ago. So these are routinely used for our population genetic study. This is very recent one. When Saddam Hussein was caught, his phenotype was totally different. So they used his son's profile and compared and identified, yes, he's the Saddam Hussein. Now I'll tell you about mitochondrial DNA quickly. As all of you are aware, this is the powerhouse of the cell, and there are a lot of activities, metabolism goes on, and it goes through mother, maternally inherited. The black one you will see, the circle is female, this is a male. I would say grandmother passing to all her daughters and sons, but the sons cannot transmit to the next generation, whereas daughters can transmit. That means all of these individuals have the same mitochondrial DNA. So why it's used? And I'll give you one basic example where this can be used. So this is one of the forensic case where they have the background of the case is that somebody has been killed and thrown in a well. It was more than a decade ago. It was done in the forensic laboratory using the STR-based method, but the DNA was very badly degraded. Therefore, they could not get the profile. Only small fragments have amplified, but not these fragments. Therefore, it's very difficult to identify whether it's the identity of the individual. There are 5 families that are claiming this deceased individual, thinking that government might give compensation. Okay. Then they have approached us, and we used mitochondrial DNA. This is one family, for example, shown here. There are 2 brothers and the father. And father, of course, the mitochondrial DNA definitely it not comes from the father. Therefore, we did not include that in the mitochondrial DNA. And this sequence and this sequence are the real brother. Therefore, they will have identical sequence. And the middle one is the mitochondrial DNA of the deceased individual. So why the mitochondrial DNA is useful here? Because that's circular in shape. There are more copy number that was be used for forensic application. And because it's intact, smaller genome, we can amplify small fragment and one can construct the complete sequence. So in this case, you can see there are 3 dots. That means these are the place where there's a difference between the real brothers and the deceased individual. You can see there's a T, time in here, and also the real brother has time here after G, right? Whereas here there's a C. Same way here there's a and A, whereas this C. Right? Here there's a between these 2 T's, again A between these 2 T's, and whereas here's a G. So with this small stretch of sequence, you find 3 nucleotide difference, then you can imagine will amplify at least 1 kb region. There will be large more variation, definitely saying that this man, the deceased person, is not belongs to this family. And it's also used for various medico-legal applications where the organ transplantation being done, and some state that both donor and recipient should be blood related. So in that sense, autosomes are used, mitochondrial DNA are used, because if both donors and recipients are maternally related. So here you see that the donor and recipients have the identical sequence, so no problem here. But sometimes, although they say that they are maternally related, but you can see there are 4 differences. Okay, then it clearly says that they cannot be maternally related. So this was one of the CBA case where somebody has been killed and thrown in one of the IIT campus, and the bones were recovered after several months. And you can see that this is the skeletal remain bone sample, the missing individual's bone sample, the mother, and this is the father's. So there are 3 panels. you can see that this bone and mother's sequence are identical. G as a T in that position, whereas father, of course, you know that father's contribution, not there's a C. Here there are two A's, both in the deceased individual and the mother. Here there's one deletion. Same way, these two are identical and this is different, confirming that this bone sample actually belongs to this family, or she's a mother of the deceased individual. Now, briefly, I'll come to tracing the ethnicity. Okay, so where it's very, very important, but it's not being used very effectively in the border security cases where somebody come from other country, do some crime, and if you find any biological material, you can actually say, you can find out whether this the individual who involved in this crime belongs to either India or outside India. You can see this is a Y chromosome haplogroup. Haplogroups are developed based on the mutation, different set of mutation present on the Y chromosome. You can clearly see that the Africans have different set of haplogroup like E, F, and G, also A and B, whereas India, we have different set of haplogroup here. East Asia, different haplogroup, Europe, different haplogroup. You can have, again, North and South America have different haplogroup. This is just one example to show that, again, they studied Jefferson's Y chromosome and they found that he actually belongs to European lineage. It must be from long back, his forefathers must have migrated from Europe. So we also have shown that the European Romas have actually gone from India about 1,400 years ago because they have unique haplogroup, which was found in India. Same way, we also shown that the Siddhi populations who are also in Hyderabad, they are in Gujarat, they are also in Karnataka. So we traced their origin and found that they are actually the Bantu speaking, Bantu is the language speaking Africans who have been brought by Portuguese traders about 15th to 19th century. Same way, the mitochondrial DNA, again, you can see large number of haplogroup, and each country and continent have different set of haplogroups. And interestingly, you might have read in the news that Prince William's, particularly his mother Diana, has come from South Asian ancestry. More details one can study in the Google. If you search, you can see more detail about that. One can also use the SNPs. We talked about STR, short and repeats, very useful for immediate relatives, father, mother, son, and grandfather, so on. Y chromosome mitochondria. Now one can also use single nucleotide polymorphic markers for same application. So this is one of our study where we use, so in this case, you can actually identify to the precise that which group this person might belongs to using about 1 million SNP markers. So we use that efficiently to look at how the Indian population structures are. We can clearly see every population clustering as a unique group. That's because we follow endogamy for the last several thousands of years. Endogamy means marrying within the group. So we did principal component analysis where you can see these are all Indian groups, and these are Europeans, Africans, Yoruban, and this is Chinese. You can see these are all Indians. Okay, so this is the Kashmir Pandit, and at this end you'll see Great Andamanis. They are in between, and this is Siddhi population because they have come from Africa and they have admixed with Indian. They are falling somewhere in between. But without seeing anybody, if you have drop of blood and if you isolate DNA and do this analysis, and if the person is European, then you will see that the person cluster here. If he's a Chinese, he may cluster here, and Africans somewhere here, right? Are Indians in this cluster? Precisely, Kashmir Pandit will be here, and other population as per this thing. So this is just one example. So we have subsequently studied large number of population. You can clearly cluster every population as unique because we acquired unique mutation because population marrying for the last 2000 years, marrying within the group, and whatever the mutation arose in each population that is restricted in those population. That's what make every population as very unique. So now I'll also give you examples of ancient DNA, but if the sample is very old, thousands of year old sample, is there any way to find out which family or which population or which country this deceased person might belong to? So this is one of the example where we studied a group of bone samples which was found in a lake called Rupkund that was about 5,000 meters above the sea level and in Himalaya. There are a large number of bone samples, and we found we analyzed those samples and found interestingly that there are three different group of samples. Of course, we studied very limited number of samples. So one group, interestingly, they are Mediterranean people, and you can see it is well spread here. So this is a group A, Indian clan, and this is a group B, Europeans, particularly French and the Mediterranean, and one group belongs to Southeast Asia. So clearly, this bone sample, we estimated this was about 1,000-year-old. Okay, you can imagine. There are many such examples. So because of time limit, I just kept only one study. And if you look at overall the application, some of them have already shown that I'll again will tell you in the coming slide. This I showed you. And this is one of the, again, important study where you can see that how the technology can be used in the medico-legal applications. So this is the samples which was the person who was been diagnosed with breast cancer, and sample has been sent to pathology lab. The report came and she thought that this is not her own tissue sample. So if she wanted to confirm whether the sample really belongs to her or not. So the DNA was isolated from paraffin embedded block, and this is the fresh blood sample obtained from her, and the DNA profile was generated and you can see that both were identical, right? So even one can go up to that level where if there is a doubt, you can actually confirm whether that tissue belongs to you or somebody else. Then it gives very, very authenticity. This I'll skip, and here this is a real forensic application where there was a rape case, and you can see this is the victim's profile, and this is a vaginal swab, and this is the person who has been caught, must be involved in this crime. You can compare this three profile and see this is a victim, and this swab, you can eliminate this and find the other one is matching with the person who has been caught must have been involved in this. Okay, so that tells that the DNA profile can be used for this application. Of course, this is the same sample used with the Y chromosome markers. As you see here, all of them are identical. And quickly in 2-3 minutes, I'll show some of the high profile cases which was earlier done by CCMB if there is a time. 2-3 minutes. Can I go ahead?
Yeah, fine. We're fine. Thank you.
Oh,
Yeah.
very quickly. That somebody might study in the news that long back, the former Prime Minister of India, Rajiv Gandhi, has been killed in Tamil Nadu, particularly Sriperumbudur, when he was touring, and she was the one who had bomb in her belt, and her sample was compared with her family and identify the identity of this individual. Of course, the samples have been collected from the belt she was wearing. And the mastermind behind the entire event, Sivarasan, who migrated from Sri Lanka, and his parent sample has been brought from Sri Lanka and has been compared. Of course, he has been shot killed in Bangalore, and his identity was done. This Tandoor murder case in Delhi, and this is one of the important cases where there was a negotiation. Actually, Dr. Lalji Singh was telling that the government that we need an institute, unless otherwise the government promise, then will not take up the case. So that's how the CDFD came into existence. So the identity of her, the Naina Sani, has been done. This is another case of the former Chief Minister of Punjab. Again, the person who involved in this crime has been identified using this technology. Many Swamijis, you must have heard that they involved in a lot of such crime. And one such case was Swami Premananda, who was basically from Sri Lanka. He had an ashramam in a place called Pudukkottai in Tamil Nadu. And in that ashramam, there are many girls. One girl escaped from there. When she escaped, she was pregnant, and fetus was aborted. The whole fetus was sent to CCMB at the time. We took the sample and we did the profile. And in fact, that was the first STR-based profile we have established. And based on the report, has been given double investment. This is one of the cases where the Singapore airline crash was in Taiwan. There are two Indians, one from Mumbai, another one from Hyderabad, and they called their parents to get the profile of theirs and send it to them, and they generated the profile of all the deceased individual, compared that body, and handed over to them. So we did that case within 24 hours. Of course, very long time. The sample came from Mumbai, night 9 o'clock. By next day, around 2 o'clock, we send the report. Sometime this also can happen, that there was a human DNA found in the Ayurveda drug. There are several drugs that have been sent to CCMB, and some drug we found as a human DNA, some not. That's because they also mix the bone, human bone, into some of the Ayurvedic drug. So that's it. Have any question? I'll be happy to discuss. Once again, my apologies for the delay in giving talk. Thank you very much.
Thank you so much, Dr. Thangaraj. It was a wonderful presentation, and thank you so much for being with us here today. Well, before we move on to question and answer from the audience, may I please invite Dr. Jivi Jagdamba, the former Joint Director DNA Division of APFSL and Head of Forensic Science in National Police Academy. She will speak about the role of DNA in civil, criminal, and wildlife forensics. Dr. Jagdamba.
Hello. Hello.
Yes, you can. We can hear you.
Yeah. Am I audible?
Yes.
Oh, okay. Thank you very much, sir, Dr. Thangaraj, sir. And a very good evening to the elite gathering. It is very noteworthy and scientific lecture by Dr. Thangaraj, sir. You all must have been enlightened by his talk. We are all aware that Dr. Thangaraj is the director CDFD, previously scientist at different levels in CCMB with vast experience in the field. We all, including Dr. Thangaraj, would remember Dr. Lalji Singh, ex-director of CCMB, and who is the founder of this genome project, along with Dr. KPC Gandhi Garu. We, the forensic people, are more aware practically of Dr. Thangaraj's great contribution in our field. Several cases could be solved because of his knowledge and requisite help in the times of necessity. I must thank him profusely for that. And we could solve many a medical legal case to the best satisfaction scientifically and also proved them in the courts of law. Justice was done in almost all the cases. This webinar on DNA fingerprinting and its medical legal applications has been set for today in the exactly right time. Medical legal cases are on the high increase in the modern times. This knowledge has to be spread across the globe by webinars such as this. DNA profiling technique is the best tool and a potential friend in investigations and casework in the forensic science field. The types of cases generally include both civil and criminal in nature, and also in cases relating to wildlife forensics. Apart from the prime role of DNA typing in the field of diagnostics, it has a very vital role in solving several civil disputes and criminal cases, and also in investigating wildlife poaching cases, and so also in determining the pedigree for seeds or livestock breeds. So for the benefit of audience and to remember some of the sensational cases which we have dealt in our laboratory, I would like to share a few. Can I share the screen, please?
Sure, ma'am, you can go ahead.
Yeah.
There should be a green button at the bottom of your screen. Saying share screen. Once you click on that, if multiple windows are open, then you can click on the window you want to present. That should be your PPT, I guess.
Yes, this is the PPT. I'm sharing.
And just press share.
Yes.
Share, that is blue button at the bottom of that mini window that has opened. On the right side.
It's not coming on this screen. You are able to see this? My window?
Not yet, ma'am.
Not yet. Okay. Yeah. I'm just saying this. Wait a moment. No problem.
Is it
okay? Okay, that's not a problem. Something is wrong here somewhere. Yeah, share screen. Yeah, now it is.
Okay, great.
Okay.
We cannot see it, ma'am.
Yeah, now? Now
not
you can
yet.
see?
No.
Yeah, not yet.
No.
Yes.
Now we can. Sure.
Yeah. Yes. In civil cases, some sensational cases we have solved. Several hundreds of cases, of course, in our previous years. Child swapping cases where yes. In one case, one person has lodged a false complaint actually with the police that his newborn child, a male child, was swapped, and the hospital authorities were giving him a female child. So such cases, there were several cases in government hospitals where this child swapping was a serious issue. The blood samples from the disputed child and the reported parents will be collected in such cases, as you know. And the DNA test confirms the true parenthood of those parents. So in this case, again, the parenthood of this Mr. Rajaya is confirmed, and bringing this light to the falsehood of this complaint actually. Like this, many cases were solved. Then in another case, one girl was missing from a family, and she was traced at a different place at a very later time, after several years. So she was grown, and she could not be, immediately recognized. So in such cases also, the parenthood of the true parenthood of the girl was also identified. So like that, there are several cases like that. In each civil dispute, we have several examples like that we could solve. In criminal cases, where the blood stains from the victim's garments or on the weapon or from the offense where the place where offense took place, all these things, they could be taken up, and the culprit can be easily recognized by conducting the DNA test of that sample as well as from the suspect and the victim person. In rape cases, in several rape cases, from the seminal stains of the victim, seminal stains from the garments of the victim or the swabs or any material which is collected by the doctor, that will be submitted in the lab. From that also, we can very easily pinpointedly identify the culprit. Even in gang rape cases, we could identify both the persons in one case like this. And in this occasion, we can just mention about this blue dress Clinton Lewinsky case, sensational case. There also, because of this DNA fingerprinting, of course, there in their laboratory, they have conducted and found. So we can say that everyone is equal before law, DNA, and all that. Right? There are several cases, murder cases and all that. This is the classic case. And in one sensational case in our area, where one family, he was the chief engineer of South Central Railway, one Mr. P.R. Seth. That was a sensational case where all the family members, 4 family members, were killed and burnt in a car. From those, bodies, we could collect samples like the internal organs or from the bones and all that. We could collect the biological material, and from their blood relatives, we could solve the case. Not only this, we could identify, trace the criminal also in this case. And because of this DNA report only, the culprit was given the capital punishment in this case. This was one of the first sensational cases our laboratory took up. So in some other civil cases like this, where family, visa cases and all that, the family relationship between the people, like in this case, one mother and son. Their true relationship was established, and the embassy of Italy, they have cleared their family visa. And in civil cases like this, organ transplantation, in those cases where the close blood relatives only should give their organ for transplantation. In such cases also, DNA fingerprinting tests we conducted in our laboratory, and we have established their relationship or establishing not their relationship. Like that, it could be solved those cases. There are several other cases. Okay. And in wildlife poaching cases, or in cases, one such classic case where the tiger skins were put to sale at one place, and the vendors duly they were arrested because it is national animal and its skin should not be sold and all that. So they were arrested, and the investigation, of course, followed. In this case, there was a suspicion that the skins on scale on the sale were not of tiger. Okay, so we have conducted several tests in our laboratory also, and we took the help of CCMB and to know to which animal exactly it belongs to. That test also we could conduct. On comparison of the exact molecular signature of vast number of known animal species which are available in CCMB in their database, they were generated and maintained by CCMB. So there we could we took their help and we could solve this case. So finally, the result is that none of them were of tigers. Meaning, like this in this case, 5 samples we have taken, and 1, 3, and 4 they were painted dog skins, and 2 and 5 were painted with cow skins. And in another interesting case, where a national bird was being cooked at one place. So police seized that material and sent it to FSL to know whether it was really of peacock or not. So here also we could conduct some of the tests, and to confirm its origin, we took the help of CCMB, where CCMB alone has the capability to undertake such cases. We could establish the identity using universal primers, and they have concluded that the source of DNA isolated from this flesh was of an Indian peafowl species specifically. So this case was also confirmed. Like this, there are several applications in forensic field. We have several applications like this. It's a very broad cross-section of disciplines, including human forensic science, diagnostic medicine, family relationship analysis, animal and plant sciences, wildlife forensics. There are various applications we know. There is potential application in the plant sciences also, to include the determination of trait markers, the purity of their traits that can be identified. And it is DNA typing has considerable potential in the animal sciences for species and individual animal identification, as I was mentioning in the previous cases also. Finally, we have this application in wildlife poaching. We have several cases. We have seen several celebrities are also included in that. So like this, in many cases, we have this application of DNA profiling, and we could solve several sensational cases. So finally, I would like to say that we have come a long way to know and identify the most primitive and the most dependable evidence. In a world which is round, the end is always the beginning. Hence, to know DNA is to know the very beginning or the end of the evidence of creation itself, which is dependable, decisive, and final. Okay, so finally, I must thank Dr. KPC Gandhi Garu, the founder of Truth Labs and the ex-director KPFSL, and also the co-founder of this genome project, for giving me this opportunity to share my views and to speak a few words after the fantastic webinar by Dr. K. Thangaraj. Thank you, sir, and thank you, one and all. Thank you.
Thank you so much, Dr. Jagdamba. This was very nicely put, the applications of DNA fingerprinting, and you spoke with utmost grace and explaining us the relevance of the DNA fingerprinting in modern age as well. So thank you so much for sharing your time with us. And now, FABA, along with, in collaboration with Genome Foundation, is proud to announce a workshop on DNA fingerprinting. And this time we are bringing a hybrid mode. We are bringing only the virtual workshop, or if you also want physical hands-on training, there is an additional fee and an add-on can be applied for. And to introduce you all about this upcoming workshop very briefly, may I please invite Ms. Aparna Kaja, a director of Genome Foundation, who comes with academic background of MS in University of Pennsylvania. She worked as a research scientist and research safety coordinator at Baylor College of Medicine and Veterans Affairs Medical Center, Houston, respectively. Ms. Kaja.
Good evening, everyone. Very happy to be part of this eminent talk given by eminent scientist, Dr. Thangarajan. I feel very fortunate today to say that Dr. Thangarajan was my guide, along with Dr. Lalji Singh, 3 decades back, that is in 1994 and 95, who taught me DNA isolations, PCRs, and also DNA fingerprinting techniques. Thank you very much, sir, for considering our request and giving this wonderful talk for benefit of the present day students. Genome Foundation is a non-profit company established under Section 25 of Companies Act, founded by a group of eminent molecular biologists, late Dr. Lalji Singh and late Dr. P.M. Bhargava, and a renowned forensic scientist, Dr. Gandhi P.C. Kaza. Besides, former Secretary DST, Dr. Palle Ramarao, and eminent medical professional, Dr. Kakarla Subbarao, and others. The present chairman is Dr. C. Rangarajan, former Governor RBI and Government of AP, and also former economic advisor to the Prime Minister of India. Genome Foundation, as you can see, you can see in the shared screen, is a congregation of highly accomplished scientists and committed public service professionals who serve the nation with very high integrity and commitment. Genome Foundation is currently engaged in, you can see the services on the screen, in genetic diagnostics services like pre-implantation, genetic screening, aneuploidy screening, carrier testing, and other screenings, also genetic counseling, DNA forensics, research, training, and skill development workshops like today's. It is working in collaboration with many clinicians, researchers, hospitals, and educational institutes around the world globally to identify and diagnose common and rare genetic disorders in the Indian population. We also conduct webinars for public awareness regularly. We provide hands-on training and skill development workshops to national and international students in the fields of molecular diagnostics and DNA forensics and cytogenetics. We also give guidance for dissertation and research projects to students. Our laboratory is well equipped with state-of-the-art equipment located at Banjara Hills, Hyderabad. You can see the complete address over there. And also the information about the phone numbers and websites and email is also given on the screen. All services are offered at affordable cost, and genetic counseling is offered free of cost by certified doctors and seasoned scientists to patients and their families. I request all present students to avail this opportunity and attend, and please tune into these workshops and hear to some adventurous cases Truth Labs and Genome Foundation has solved in real life using techniques like DNA fingerprinting along with other molecular diagnostic techniques, because it's a great opportunity. As a sequel to this webinar, Genome Foundation will be organizing series of workshops, both in virtual and offline mode, starting with a virtual DNA fingerprinting shop in the last week of October 2021. The details of workshop will be given by Ms. Gargi Desmukh, scientist in charge and DNA expert of Genome Foundation.
Thank you so much, ma'am, for the introduction. Everyone, welcome. And I'm so glad I could hear Thangaraj sir and Jagdamba ma'am speak about DNA fingerprinting. It is an honor to hear them and a pleasure to hear about what they've said. In continuation with what they have said, we are going to be conducting a 3-day virtual DNA fingerprinting workshop along with FABA, where we want all the students and participants to learn more about DNA fingerprinting. So this is going to be conducted in the last week of October, from 25th to 27th of October. The focus of the virtual DNA fingerprinting workshop will be on what is DNA fingerprinting, the basics, the principles, and the protocols that are used. We shall also go into details about the applications and uses of DNA fingerprinting. So the types of cases that Genome Foundation, Truth Labs have handled, how they have helped society, how they've given closure to families, and how it has helped people get, closure in terms of legal as well as medical conditions and cases. We'll also be having case studies presented by eminent scientists, and they'll be talking to us about their sensational cases in this workshop as well. For all those who are interested, be they students, PhD scholars, faculty, or industry professionals, we request you all to join in, sign up and join in for the virtual DNA fingerprinting workshop. All the details regarding the workshop, the queries can be asked on the website listed over here, or the email address or the mobile number, or you can also talk to anyone at FABA, and on their page also it will be available. The flyers will be circulated shortly. We all welcome you all to join in and enjoy this 3-day workshop. Thank you.
So thank you so much, Dr. Gargi, for introducing our audience about this upcoming workshop. And now, with pleasure, I have already posted the link for registration of the workshop in the chat box, everyone. So once again, this is a virtual 3-day workshop, which requires commitment of 3 hours per day, 10 a.m. to 1 p.m. On 25th, 26th, and 27th October. And also, if you are keen on learning these techniques in hand, we have an add-on option where you can physically visit Genome Foundation, and you'll be guided by scientists like Dr. Gargi, and to hone your skills on these techniques via hands-on training. That would be 5 days in total. Every information of how to register for that workshop is given on this website, or you can also write to me at academy.faba@gmail.com. I will take this opportunity to also let you know that there is another workshop, which is workshop on biosafety and biosecurity, and the information of that as well is present on our website, which is biofaba.org.in. And so definitely visit the website for all this information and register. One more thing, as you all know, FABA members are entitled to 50% off on all the courses, on all the workshops that are launched by FABA. And hence, if you're a member, if you want to avail this, you will be enrolled in this workshop at 50% discounted price. And so let us know through email if you're keen on doing that, because then we will provide you an additional link where you can pay the discounted price. And now for the people who are keen on getting the certificates, right? Please, in future, do not prompt us in chat box again and again about the certificates. This is a kind request. In the meantime, you may focus on gaining some knowledge, right? If you were that eager to learn even 5 minutes of what the speaker is saying, you would find yourself on the other side of the road where you're more successful in learning and in being expertise at a topic. And so I will now post the link to the certificate. Please go ahead, fill in, and the platform is now open for a healthy discussion of question and answer with the speakers. May I present the first question that I found in the chat box? This is from Neha. She says, I want to ask how this technique can improve the area of personalized medicine. So how this technique of DNA fingerprinting can improve the area of personalized medicine. Dr. Thangaraj, could you please share your perspective?
Yeah, so I'll give a little bit background about the personalized medicine before going to detail. So you know that every medicine we take, there are genes which metabolize. Okay. And the mutation in those genes decide whether you get therapeutic effect, no effect, or adverse effect. Right? So one has to look at the variation present in that particular gene, and sometimes that also determine how much of dose is required for you. For example, I can take vitamin K epoxidase reductase complex 1, that is a target gene for warfarin. Okay, and in US earlier, the dose has been determined with very mild and gradually being increased, and they optimize the drug, how much they have to give. And sometime later, they found vitamin K epoxidase reductase complex gene is the target gene for warfarin. They sequenced the gene, identify the mutation, then they went back to the medical record and they constructed the haplogroup, and there are 7 different haplogroups. And haplogroup 1, which has a certain mutation. Haplogroup 2 is not very significant. Haplogroup 3, 4, like that. And haplogroup 6 or 7, that has a lot more mutations compared to haplogroup 1. Now they took the medical record and seen how much of warfarin has been optimized to those individuals. And they found that haplogroup 1 required 2.9 milligram of warfarin per day, right? Then haplogroup, I don't remember, 6 or 7, and they found the record shows double dose, 6 milligram of warfarin per day. Now you see that the dose has been optimized without looking at anything, gradually optimized. Now, when they looked at their genome, and that shows that a certain mutation requires less quantity of warfarin, and on the other hand, different set of mutation require double the dose. So now, if anybody wanted to treat with warfarin, then from the day 1, they can sequence, identify the mutations, and decide how much of warfarin one has to be given. Otherwise, it may take several months to optimize. Okay, so I hope that it's clear. So basically, this is also a fingerprint. So basically, using the DNA variations, and you are optimizing.
Thank you so much, Dr. Thangaraj. There's another question for you. Is there any maximum extent of degradation of a biological material such that no DNA can be further extracted from it?
No, it's not that the degradation goes with a well-defined way like what we use the restriction enzyme. Restriction enzyme we use, there is a restriction site, it cuts exactly at that position. But the degradation happens randomly. Okay, so you can see the random and range of fragment. You might see degradation to extent that 100 base pair, right? From 100 base pair to, for example, there could be 1 or 2 molecules at either 1 kb or 5 kb, for example. And in between, there could be 5 copy, 10 copy, or something. So for the sake of telling you, 1 copy of 1 kb region, 200 copies of 100 base pair, or in between rest of it. And if you try to amplify more than 1 kb region, you may not amplify anything. Right? If you try to amplify less than 1 kb, 800, there could be few copies, and you may have to re-amplify. But if you want to amplify 100 base pair or 200 base pair, that can amplify effectively. Okay, so that's the answer. If you still not clear, you can ask again.
Thank you so much, sir. Are there any more questions? Please go ahead and type them in the chat box. If any follow-up is required from the previous questions, you're welcome to ask. Okay. So I think, sir, I think we're done with the question and answer round. So may I please now invite Professor Redana to deliver a vote of thanks to the speakers today. Oh, sir, there is a question that appeared. I'm so sorry.
Okay.
Whether digital PCR can improve this technique further and how?
So digital PCR, you are just predicting using your designing a primer, try to amplify, and it will give you some kind of prediction. It's not really that then once you predicted that you can go and do the experiment.
Right. So there's another question. Is there any concept called touch DNA?
Yes, that's a method called touch DNA,
Okay.
which exists.
Could you like throw some light on that?
So it's not like you're taking one touching and the DNA. So I showed in the initial concept like somebody who has taking tea or coffee or smoking cigarette or something like that, where they unknowingly they leave some of the biological samples, right? So taking out those materials and try to use for DNA fingerprinting. Okay, so that's it.
Okay, so thank you so much, sir. And may I request the audience to please sign up for the workshop. Why? As you see, one of the questions in the chat box is RFLP means what? Please explain. Now, workshop is the best area to get a wonderful idea about what RFLP is and how it is applicable. And so definitely take this opportunity to sign up. If there are any queries or trouble with the registration, get back to us, write to me on the email academy.pharma@gmail.com. I'll be happy to help you. And now, as the time is limited, may I please invite Professor Redana to deliver the vote of thanks.
Thank you, Dr. Thangaraj. First of all, apologies for the confusion. We should have approached you in the morning itself. But anyway, that became inevitable. Despite that, within the short notice, you could come back and then give the talk. Really, I'm sure every participant here would have been enlightened with your talk, starting from the history of DNA fingerprinting and how the DNA fingerprinting started in India. And the father of DNA fingerprinting, we can call Dr. Lali Singh as the father of DNA fingerprinting and diagnostics, and who was responsible for the establishment of the institute for which you are now the director. And so you have started with the basic fundamentals of DNA fingerprint and very clearly elucidated the technology behind and many of its applications, not only in forensic, but also in tracing the history of mankind, a revolution of man. And several of your publications are really landmark publications, and we are all proud of your contributions and Dr. Lalji's contributions. And Dr. Lalji was responsible for the Genome Foundation establishment, and the Genome Foundation, after Lalji Singh, Dr. Gandhi has taken up the responsibility and passing on the benefits of genome technologies, including DNA fingerprinting, to the common man. And very recently, they have even entered into the skill development program. That's where FABA is really proud of, very happy and thankful to Dr. Gandhi for joining us in offering the skill development programs. not only online, but also hands-on. Hands-on may not be for many, but it could be in batches of 5 or 10 per batch. And so let me also thank all the participants who could bear with us for the delay in starting the session. And that became inevitable. And thank everyone. And Dr. Meenakshi and Dr. Nidhi, both of them have really coordinated well and covered up this lapses from our side. Thank you, one and all. And I must say that Dr. Thangaraj has agreed to give introductory lecture for the workshops that are going to be conducted on DNA fingerprinting whenever it is being done. Thank you, one and all, and maybe see you back for the workshops.
Thank you so much, Professor Redanna. And I would like to take this opportunity to thank each and every one of you who's been able to join us today. And you were very kind to return at 4:30 for the webinar, as mentioned by Professor Redanna. And thank you so much, Dr. Thangaraj and Dr. Jagdamba, Ms. Aparna and Dr. Gargi. It was wonderful having you all on this platform today. And may I please request the audience once again to please register for the workshop and take the benefit of the knowledge from the experts, learn about the applied sciences. That is where the future is, where the science currently is and where the future lies. And so please attend the workshop to understand the applicability of DNA fingerprinting. Definitely reach out to me in case of any queries. Do not forget to join us on Telegram. Everybody, we do not want to give you any pains of following us everywhere with the updates, and so we bring you the updates ourselves so that it's just prompted on your phone. So definitely join the Telegram channel. And with this, I will be looking forward to having most of you at the workshop. Thank you so much. Thanks for joining, and we'll see you very soon. Bye-bye, everybody.
Bye. Bye, sir.
Bye. Bye.